we're going to go ahead and take a look at chromosomes, genes, alleles, and mutations as an introduction to genetics. So let's start off by talking about how DNA is actually packaged. So in the center of all of your cells, there's a nucleus, and your nucleus and every one of your body cells contains 46 chromosomes, and each chromosome looks like this. Each chromosome is made up of two sister chromatids. We'll talk more about that later, or go review mitosis. We're going to be learning about meiosis pretty soon, so we're going to revisit this as well too. But the chromosomes are kind of like a spool of yarn. If you were to pull on one end and stretch it all the way out, eventually you end up with DNA. The DNA is actually packaged by being coiled around these little proteins here, which is coming up in the next slide. And after they get packaged like this, they get coiled again and again. It's called supercoiling. And this helps package all of your genetic information into each of your tiny little cells. Keep in mind that every single one of your body cells contains your entire genome. You're going to see that word coming up in a little bit as well too. So your DNA gets packaged into these chromosomes by being coiled up super tight. And we're talking specifically here about eukaryotic chromosomes. We are eukaryotes, and the other eukaryotic organisms are plants and fungi and protists. But here's a close-up diagram of how that supercoiling is actually happening. I told you that the DNA is wrapped around these proteins, and these proteins are called the histones. They're called histone proteins. And every core that looks like this actually has 8 histone proteins, 8 histone molecules. And each one of these little subunits right here, so when I'm circling with my cursor right here, is called a nucleotide. So we can say that DNA is wrapped up tightly with nucleosomes. One nucleosome is basically 8 histone proteins surrounded by a length of DNA. This continually happens to all the way around. Wrap up the DNA really, really tight. A gene, so when people talk about how my parents passed my genes to me, I am what I am from my parents. A gene is a basic unit of heredity. Chemically, what a gene is just a length of DNA. But because that is more important than just the letters that the DNA is made up of, it actually codes for something. So we give it a separate name. It's a gene. A gene is a specific length of DNA. It's a basic unit of heredity. It's a segment of DNA on a chromosome that describes how to make a certain protein. So if you've already learned about transcription and translation, you'll understand that a gene can get transcribed into mRNA and then translated by a ribosome into an actual protein, a sequence of amino acids that is a protein that has a certain function. And proteins can be anything from enzymes to hormones to antibodies to structural proteins, like collagen in the skin and in the hair, basically. And genes are located at specific locations. It's easy to remember here. A location is basically a locus. More than one would be 2 or more loci, L-O-C-I. So on a chromosome, there is a specific locus, and at that specific locus, there may be a specific gene. So we're going to be looking at that when we're talking about genes for eye color, genes for particular diseases, genes for producing hemoglobin, for example, will all be at a specific locus on a specific chromosome. The thing is that all humans, we all have 46 chromosomes in every single one of our cells, and we all produce the same enzymes. And there may be some variations, but in general, things that we really need, like amylase in our saliva to break down starch, we all have a gene to make that enzyme called salivary amylase, and that gene is located in the exact same place for all humans. Okay, that's why men and women can have babies and they can produce functioning children, because those genes are on the same location, and that mix of genes will determine the specifics of the baby. One more thing is called the genome. The genome is the whole of the genetic information of an organism. So we can talk about the human genome. That's all of the genes combined in all 46 chromosomes of humans. You can talk about the fruit fly genome, or you can talk about the flatworm genome. It's specific to each organism, or even for plants and bacteria as well. Okay, the E. coli genome is all of the genetic information that's found in the E. coli, for example. Okay, here's a word. Remember how to spell it. It has 3 L's in it. You can pronounce it allele or allele, depending on where you are in the world, I'm gonna probably switch between them because I'm inconsistent like that. But an allele is basically a different form of the same gene. So you can talk about a gene for eye color, which is located at a specific locus, but that gene could have different forms, and that's why you can have different types of eye colors. Eye color is actually more complex, like height and propensity for obesity, something like that. But if you talk about other types of things like, I don't know, the presence of freckles or the absence of freckles or the presence of mid-digit hair. I'm just looking at my hand right now. If you have some hair that's hanging out on your mid digits, that's also controlled by a gene, but that gene can have different forms. One form results in hair being there and one form results in hair not being there. So we're just going to use eye color as a simple example here. And an allele is any of the different forms of a particular gene that occupies the same locus as other alleles of the same gene. So the gene is always referring to a particular position on a chromosome, but it could have different forms. And those different forms are just, chemically speaking, a few differences in some of the letters there, maybe only one letter difference. So if you know that DNA is made up of A's, T's, C's, and G's, then you may realize that two different alleles for the same gene just might have a few different letters that are different. So it differs from other alleles by only a few bases. And these alleles are represented by letters. So when we talk about genetic Punnett square crosses determining the genotypes and phenotypes of the kids, you're going to be using a lot of letters like big A, little a, big B, and little b. So for example, here are two chromosomes, two of your 46 chromosomes, one you got from dad, one you got from mom. Notice that the locations of the genes are the same on the exact same size chromosome. So in this case, the eye gene is located in the same place, the hair gene located in the same place, tongue roller or not tongue roller, the gene is located in the same space. This is kind of a joke, not so smart gene versus smart gene. The point is the genes are located in the same spot, in the same locus, but the form may be different. This dad may be giving the big B form, while the mom may be giving the little b form. Big B meaning brown eye, little b meaning blue eye. So what will the kid get in the end? Well, this depends on dominant recessive relationships, which will come up later. But it's important for now to understand that alleles are different forms of the same gene. Different forms of the same gene. You don't have to get different ones from both parents. In this case, let's see that both dad and mom are contributing a big T, or the ability to tongue roll, into their kids. So it's highly likely or almost statistically guaranteed that their kid is going to be able to roll their tongue as well because they're getting the same form here. Okay? The number of chromosomes we've mentioned already. Most human cells, or all human cells, should have 46. There are exceptions if there's various types of genetic diseases. 46, that's 2 copies of 23. So 23 you got from mom and 23 you got from dad. This number we call the diploid number. When cells have 2 copies of chromosomes, we say that the cell is diploid. There are some organisms that have 3 copies or 4 copies, and that gets very complex. But for us, we're humans, it's easy for us to remember that our diploid number is 46. So that's all our body cells have 46. The only cells that don't have 46 are those cells that are involved in making more people. That would be sperm cells and egg cells. That makes sense because when a sperm cell and egg cell are going to combine together, you have 23 in one and 23 in another, and that makes a total of 46. So n equals 23 is considered the haploid number. Think of haploid rhyming with half, 23. Diploid sounds like double, so we can say n equals 23 and 2n equals 46. All your body cells or somatic cells have 46 chromosomes. Sperm cells and egg cells have 23. And that makes sense to make a normally functioning human being. Okay, that was a short introduction. The next video we're going to continue talking about mutations that can happen, and one specific example of a mutation which results in a well understood disease called sickle cell anemia. Stay tuned.